Zoom by Rothenberg from the California Institute of Technology. And paradigm shifts in molecular biology and immunology, witnessing non-Kuhnian transformations.
Hello, and thank you very much for letting me participate from far away. I hope this is going to work out all right, and let's just see if I can make it work. I felt that as a practicing molecular biologist who had many of the formative years of my own career during a period which is really a golden age for conceptual innovation in molecular biology, it might be interesting to just sort of bring in my grunt's eye view of what it actually was like to go through transformations in knowledge and to see the extent to which they don't conform to at least my impression of the Kuhnian worldview. And so I'd like to share this with you if you think that this would be interesting and appropriate for the meeting. Well, let me see if I can actually get this connection to work from far away. So I'm going to switch over to this. Actually, I can't do that. I have to first share it, I believe. First, I have to share this. So if I'm sharing the right thing, I think that I'm sharing the right thing at the moment. And let me then switch it over to presenter view so that you can see it. Okay, all right, can you see my screen properly?
Yes. Yes.
Can you see it?
Yes.
I don't hear anyone else except myself.
Yes, I can see it.
Great, wonderful, thank you very much. Okay, so this is paradigm shifts in molecular biology and immunology, and my experience witnessing what I would say are non-Kuhnian transformations. So one of the questions is, what is meant to be the relationship between great advances in understanding and what Kuhn famously called paradigm shifts? And one question is, what is the role actually of novelty? Obviously, novelty is playing a role in changing our understanding of something. What's the role of technology advance? And particularly, is there really a distinction between normal science, which Kuhn was pretty disparaging about, and revolutionary science? And I guess one question is the definition of what's normal and what's revolutionary. How much resistance must a new idea face in order to qualify as a paradigm shift? And how, in terms of impact, how many other people beyond the discoverer must have their own research changed in order for discovery to qualify? So let's talk about what I actually saw. This is a short summary of just things that happened in molecular biology between the discovery of the structure of DNA and when we finally understood that many aspects of the paradigms that were developed as the progress of this field went forward actually had to be discarded and replaced with new paradigms because of the pace of discovery. So we're talking about the structure of DNA was only discovered in 1953. Crick put together the picture of DNA to RNA to protein as an information flow, which he helpfully christened the central dogma over the next few years. And meanwhile, others were discovering the genetic triplet code for protein synthesis, which is really the way that the genome encodes protein structure. In parallel, the principle of differential gene regulation and its molecular basis was being worked out while the triplet code was being established. And the very important point was the organization of protein coding information in the genome and its collinearity. And this really led up to maybe 1966. Within 4 years, there was the breaking of the central dogma that RNA, and by the demonstration that RNA can template DNA. And then at the same time, that opened the door to gene synthesis in vitro, restriction mapping, genomic cloning, which are technological advances, but which totally empowered major conceptual advances, DNA sequencing being invented, and then breaking of the collinearity rule for eukaryotic genes and eukaryotic viruses, and breaking of the genomic invariance rule for immune cell receptors. So a number of these revolutions happened while I was a graduate student and happened on the floor of the MIT Cancer Center while I was there. And what you can see in these two pictures from 1975, I'm the little green arrowhead in the two pictures. And the people under the yellow stars are Phil Sharp, Sue Burgett, and Claire Moore, who are the people in MIT who discovered splicing. In the lower picture, it's just Sue Burgett and Claire Moore. And so these were people that I rubbed shoulders with every single day before, during, and after their discoveries. And what I'd really like to ask is, this period where there was tremendous advance going on, was this normal science by a Kuhnian definition? One important point was the rapid and progressive technological development, and focusing on the question of how DNA and RNA molecules related to each other in sequence organization and in sequence identity. And there was constant invention, partly because the people who were doing this work were as much biochemists and biophysicists as they were molecular biologists. So there was a constant attitude about improving the enzymology, improving the hybridization reaction measurements to determine state transition kinetics. And the enzymology of DNA replication in particular was very heavily exploited. One other feature that really owed a lot to the physicists in the field was our constant use of radioactivity as a marker for newly made. It's a dream marker because it's highly quantitative. It can be related directly to the molarity of the product. So you actually know the physical amount of product by putting a vial on a scintillation counter and counting the counts. And easy to detect. And so it made it possible to distinguish newly made from non-newly made molecules and also detect their physical properties. And that turned out to be incredibly important. There was a rapid development in the ability to use sizes of nucleic acid polymers as position indicators in genetic code. In other words, determining what a sequence was, what two sequences were related to, by how far apart they were in a molecule that you could size. And so that meant that the development of good sizing techniques that allowed parallel comparisons was very important for this. And actually defining landmarks against which you could measure lengths in a meaningful way was critical. And the great advance in this is the discovery of restriction enzymes from bacteria, which cut at simple sequences that were relatively rare in the genome, and so you could make discrete-sized pieces of DNA with defined endpoints. And the sizing I've already talked about. Another incredibly important advance was the discovery of nucleases that would cut back single-strand DNA or RNA, so you could convert a hybrid of two strands, one shorter than the other, or the base paired part being shorter than the other, converting it into a length relative to the starting length, and that would actually tell you how much of a duplex was, how large a fraction of the starting strand was actually involved in a duplex. And electron microscopy played a very large role in this, as you'll see in just a few more slides. And restriction and fragments, which people initially were seeing really as a way of mapping breakpoints in the DNA caused by enzymes, when they realized the biochemistry of the ends of those restriction enzyme fragments, they realized that they were complementary, and that immediately led to the invention of nucleic acid cloning, which completely transformed our ability to do molecular biology. Now, another aspect of this, which many people are not aware of, is that it was extremely important that in the 1970s especially, there was a huge push to work on viruses, and this really had to do with Nixon's war on cancer and the special virus cancer program. It had been recognized that certain viruses could give rise to leukemia. And people were working on viruses then not only for the kinds of diseases that they cause, like the cold that I'm suffering from right now, but also for things like leukemia. And this was incredibly important because it was the best access to mammalian molecular biology that anyone in the field had over this critical 10-year period when technology was advancing very fast. Why were they so important? There were technical and there were also conceptual advantages. Technically, they have very small genomes, so the fairly crude tools that we all had available to ourselves in the 1970s were adequate to deal with a genome the size of a virus. This was usually somewhere between 7,000 base pairs and maybe 30,000 base pairs. That's a pittance compared to our genomes, which are 3 billion base pairs. But this was, and E. coli, which is 10 times, which is 1,000 times smaller. But this is 30,000 base pairs. You could fit a lot of genes into that. And the virus, as part of its life cycle, would actually replicate these genomes and put them in little packages, which you could isolate physically. And so it was the most lovely Christmas present for the investigator that it would get these little genomes delivered in packages. The point was that all the viral-specific proteins that the virus depended on for its replication had to be encoded in the viral genome. And so in this little tiny defined package, there was absolute logical clarity. Everything had to be there. The proteins that were coded by the virus were the virus's important proteins. And because there were defined time courses of infection, you put the virus onto cells and you wait 3 hours, 6 hours, 9 hours. You knew exactly what the virus was doing at that point, and you could separate different classes of genes, early, middle, late genes. So that was these were technical advantages. But a very important conceptual point was that viruses are not cells. Everyone knew this. And so they had a license to be viewed as different from what everyone thought were the rules for cells. In other words, they were free to be paradigm breakers. And by working on viruses, you were basically opening yourself to a paradigm shifter, and I'll talk about that a little bit more in just a moment. On the other side, the flip side of this, was that they clearly depended on mammalian cells in order to grow. By definition, a virus cannot replicate itself. It needs to infect a cell and use the host cell's machinery. And so that meant that the gene expression of the virus, even though it basically was viral proteins coded by the viral genome, and viral genomes often being replicated by viral enzymes, nevertheless, the gene expression would have to observe mammalian cell rules for how you actually have RNA translated into protein. And so you could use that to create new paradigms. You could test them out on the virus, which was free to be different from a cell. And then you could just ask if the cell might have something like what the virus was doing, in fact. And so, for example, there were some very important rules about mammalian cell biology that were actually first discovered in viruses, originally labeled as being odd things that viruses did because they were weird, and then almost immediately afterwards being discovered to be in mammalian cells as well. And one of them is only one ribosomal initiation per messenger RNA, and only one, and a special structure on the 5' end of the RNA. Now, this slide, which just rapidly advanced itself, is important because it goes into a little bit more detail about what aspects of virus enabled them to be paradigm breakers. It was specifically with respect to the genome and the central dogma that DNA is the storage form of the genome, that RNA is copied from the DNA in order to make a messenger that's used as a template for making protein. Viruses were known in the 60s to have many different kinds of genomes, not just double-stranded DNA. There were single-stranded RNA genomes in some viruses, like polio, same polarity as messenger RNA. The genome could act as a messenger RNA, but it was also the genome of the virus. And so it replicated, but how? It didn't have double-stranded DNA. Who was replicating it? There were other viruses that were single-stranded DNA, the opposite polarity to messenger RNA, which meant that when they first came into a cell, they could not even be translated into protein. They were just an RNA molecule. What happened to that? How did it replicate? How did it encode proteins? And then there are double-stranded RNA viruses. And then the easy ones to understand would be the double-stranded DNA viruses, which have the same genome structure as our own cells. But all four of these things worked as genomes, and therefore at least three types do not follow central dogma. And that is obvious from the outset, right? So you know that the central dogma is imperfect with respect to viruses. Okay, so let's look at how that actually impacted on these revolutions. And so let's go back to this, and we'll look specifically at one thing which happened exactly in the middle of my graduate career, which was the breaking of the collinearity rule for eukaryotic genes and their viruses. And this green bar sort of covers the years when I was a grad student at MIT, and basically the advances in knowledge and technology that took place at that time. So where did the idea of collinearity come from? It actually came from a beautiful, sophisticated, difficult tour de force that was done in the 1960s, applying the logic, the rigorous logic of genetic mapping, which is a very abstract idea, by recombination, and then attempting to determine when you had different recombination points in a gene coding for the same protein, how did that relate to the position of the mutation in the protein? And this, the idea of genetic mapping was used in fruit flies before the structure of DNA was even known. Bridges made the first genetic maps of Drosophila, relating them to chromosomes, even though they didn't even know what in the chromosome was carrying the genetic information. And it was used extremely powerful in the 60s for bacteria and bacteriophages. A very important thing was they identified a class of mutations that made proteins not only be defective, but caused them to truncate prematurely. These were called amber mutations. They were very important because if you had an amber mutation, you knew the protein was going to stop at that point. And so then if you actually did the recombination mapping of a series of different amber mutations, you could then relate that to the sizes of the proteins that were made and ask if they were a nested set, if they were non-overlapping, or what were they. And in '64, that was a golden year. Two different groups, Saragai, Stratton, Brenner, and Bowley, and Charles Yanofsky et al., both came up with essentially the same kinds of evidence. And they showed that if you have a recombinational map, which is here shown, let me see if I can get a pointer for you. Here's a laser pointer. Okay, so if you have a recombinational map with mutations mapped based on if you had a crossover between this mutation and another mutation, was a third mutation between them, or was it always going with the right one, or was it always going with the left one? And so that's how the mapping worked. They related it to the lengths of the proteins that were made. And they actually found that the order in the recombination map was the same as the nested set lengths of the proteins, and the peptides that were contained in the proteins were a nested set. Now, what this meant was that the beginning and end of the gene encoded in DNA should match the extent of the RNA if it was following this rule. And this had huge implications for the way people imagined they could, that a genetic or material is organized, and imagining that you could actually read the genetic code directly from the sequence once you had a sequence, because it would actually give you the sequence of the protein. But the question is this true everywhere? And the bacteria and their phages in which this was shown in 1964 have little, efficient, compact genomes with very little waste between protein coding genes. And it was already known in the 1960s that multicellular eukaryotes like ourselves have much larger genomes. Is that necessary? Is it garbage? What is it? And it was also known because of the hybrid nucleic acid reannealing kinetics that the biophysicists were doing, that there are a lot of repeat sequences in the genomes. And you were talking about Eric Davidson in the previous talk. A critical person in this field was Roy Britton, who did a lot of the theoretical work on nucleic acid melting and reannealing kinetics and relating that to the copy number of a given sequence in a sample of DNA. And it meant that some of the DNA that you thought you had in a genome was actually just multiple copies of the same thing. It wasn't even extra genes. And so what was that all about? And was it sensible or was it nonsense? And also it was known that multicellular eukaryotes made their RNA initially in very large molecules based on biosynthetic labeling, much larger than the messenger RNAs that would be found associated with the protein synthesizing ribosomes in the cytoplasm. And also these large RNAs were labeled rapidly when you put in a biosynthetic radioactive label, whereas it would take time before the label would start appearing in the messenger RNA. So the question was this actually a precursor-product relationship? And in that case, where were the extra sequences in the large nuclear RNAs going when they made the small messenger RNAs? In other words, we really didn't know, after this collinearity of the genome analysis, we still didn't know how multicellular eukaryotic genomes were actually expressed. So we knew that this was a problem, and new technologies had to be developed. And when you read the papers that people wrote at the time, they were very open in those days about talking about their struggles with the shortcomings of the technologies that were available. There was no sequencing. Base compositions had to be inferred from thin layer electrophoresis and chromatography. You can't even imagine somebody doing this today. There was no cloning. There was no way rigorously to isolate a single sequence from a mixed population. But what people did was they used what they had, which was strong theory and practice of using nucleic acid hybridization in ways that people have forgotten how to do now. And also critical, the advances in restriction enzyme mapping of DNA molecules physically, and also high-resolution gel electrophoresis for size separation, which really just came in as I was halfway through my PhD. And that actually enabled people to develop technologies like the ones shown here. This is actually one of my own heteroduplex analyses that was published in 1978. It actually shows splicing in a retroviral RNA. But the point was that this is actually a hybrid of a long DNA strand and a shorter RNA strand showing the position of these regions. And you can see that this stalk is a duplex. And this kinkier loop is actually a single strand. And you can relate these lengths very precisely. And you can also relate them to another gel electrophoresis technique, again for relating one long DNA strand to RNA strands, which may or may not be encoded continuously on the genome. If they're only from here to here, they would look like the length of the DNA that was hybridized with the RNA was just a pure straight line, just purely flat against the RNA. But if the RNA is not continuous with the DNA, if it actually started in one piece of DNA and then the rest of it came from another part of the DNA, then you'd have a loop of the DNA sticking out, which you could cleave with a nuclease that recognized the single-strand DNA as being vulnerable, and you could actually see these different bands and infer what their structure was. This was very important, and at this time, this was absolutely revolutionary. And so, in 1977, two different labs, Phil Sharp's lab at MIT and Rich Roberts' lab at Cold Spring Harbor, simultaneously discovered splicing. They proved it definitively using heteroduplex analysis. And these are just examples of the pictures. They're very hard to look at unless you've actually looked at a lot of these. But the keys down below show the loop structure. And here's an interpretation of the loop structure that Phil Sharp very helpfully drew for his Nobel lecture. So it's very amusing for those who are interested in the sociology of science that 3 of the 4 lead investigators on these papers were female, none of whom were represented in the Nobel Prizes that were given for this work. That was the 1970s and 80s for you. the point was that the recognition for the significance of this work was instantaneous. Both the Sharp Lab and the Roberts Lab presented their work at the Cold Spring Harbor Symposium in the early summer of 1977. By the time the papers from that symposium were collected 8 months later and published in a book form, not only those 2 papers, but 4 other papers from speakers at that meeting had added sections about splicing of RNA, which they also found. The point was that, I probably went too fast through this, that although the RNA encodes this protein called hexon, which maps to this region of the adenovirus genome, the beginning of the RNA comes from a different part of the adenovirus genome. Actually, it comes from 3 different pieces of the adenovirus genome before it even gets to this. And although these are far apart in the DNA copy, they're actually physically connected in the RNA. It's been broken and joined. Okay, so this has huge implications. For anyone who thinks that this is just textbook stuff, this is actually a continuing problem, many years later when we've actually got full sequences for the genomes, not only of humans and mice, but for many, many other organisms. We still cannot read the genome without empirical evidence of RNA structures because the genome is actually gobbledygook. And with a gobbledygook is that there are actually words in there, but there are random spacers of nonsense sequence in between them. And unless somebody tells you which part of this line of text actually represent the words, like in the section diagram here, you can't read them. So this is a perfectly reasonable sentence. The effect of heredity on responses of individuals to drugs is a topic of exceptional interest. That's perfectly reasonable. But you have to know which letters to remove in order to get that. And it has a very big implication for medical genetics, as many people are very aware, because unless you know where a missense mutation is that changes a letter, you don't know whether it has an effect on the actual words, i.e. the protein coding sequences, or not. The same single letter change can either have no effect, or it can have a very big effect and change who is a property of what. And so you don't know that without actually having the RNA structure. Now, this was actually quite a large discovery. And it's really a mistake to trivialize it by the fact that this was a continuous process of scientific discovery. And so the question is, why do I call this non-Kuhnian? And one aspect is that basically, even though Francis Crick vainly called his rule of thumb a central dogma, it wasn't a dogma. It wasn't comparable to having 1,400 years between the Ptolemaic model of the solar system and the Copernican model of the solar system, with a little help from the church to enforce a failure to progress forward. Instead, these are discoveries that were made really quickly one after the other, by people who are actively changing the technological foundation of their field year by year by year, specifically to dig into these questions with more vigor. And this may be something that Kuhn would call normal science, but it was certainly an absolute engine of conceptual advancement. Now, I wanted to switch gears to another thing which is also extremely interesting, but on my screen I unfortunately can't see what is happening in terms of time. Could someone give me a time check on this?
You have 10 additional minutes.
Okay, I'll just go forward and see how fast I can go.
Including the session.
So, one point which most molecular biologists would have agreed on was that cells in an organism have the same genomes. And that basically this is a very large part, not only of the central dogma, but just our understanding of what a genome is in a cell of a multicellular organism. And so another quite radical discovery of this period, almost identical time to the splicing discovery, was the discovery that immune cell receptor genes normally undergo heavy somatic mutation in the cells that are going to use them for their business of normal, healthy organisms. And the problem with this was that people had realized since the early 60s that antibodies, which are called immunoglobulins, the proteins within a same individual, the antibodies have different amino acid sequences. And people could sequence proteins, and they found that especially the N termini of the proteins were quite different from each other, even though the C termini of groups of these proteins would be basically identical. It was very difficult to look at this in the products of normal B cells because there are on the order of 100 million different B cells all making different immunoglobulins in our bodies at any one time. But when you have a B cell leukemia, the leukemia is clonal, and the leukemia is chugging out lots of immunoglobulin proteins, and they're all the same kind. And so basically different leukemia patients or different leukemic mice would give you different clonal cell lines that would produce different immunoglobulins, and that was something that people could sequence. And they found that there was a variable part of each immunoglobulin at the N terminus and the constant regions at the C terminus. And the question is, how does the genome encode this? And there's a number of problems. The first is the level of diversity was astonishingly high. And so the question is, how is the genome encoding so much diversity? Are there actually literally hundreds or thousands of immunoglobulin genes side by side in the genome? The other thing, which was structurally very hard for people to figure out, is even if there were thousands of genes, how could the genome do this while ensuring that the constant regions all stayed the same? And this problem was formulated before anyone had any idea of splicing. So if you think of these as collinear units, from the variable part right smack into the constant unit, how one part can be completely different, one copy to the next, while the other part's identical, was not really easy. And it was even more complicated than that, which I won't talk about. But there were two basic ideas. One was the germline diversity, assuming that the genome somehow managed to include these huge arrays of these genes that code for the different immunoglobulins. Was there really enough genomic space? Can anyone count the number of genes and see if it's really big enough? How do you regulate them correctly so that the B cells are expressing them if there are really thousands of these genes? And the alternative hypothesis was a somatic mutation hypothesis, which is that there'd be just a small number of these genes, but that something about being a B cell made the cells start to mutate themselves, and that there was a mutator gene in there that would encode an enzyme that would start to vary the sequence of the DNA. And so the question was, how come then only part of the molecule was mutated and the other part was completely protected from mutation? And it became even stranger because there are some immunoglobulin responses that are extremely similar among different individuals. Some B cells that in mice respond to a particular phosphorylcholine antigen with almost the same antibody response in different mice. How does that make sense if you're randomly mutating in everything? So there was a third model, which was of recombination in B cells, but this was considered to be really strange. And so the question was really, how do you even conceptualize this? And I think that it was made a worse conflict because of the central dogma, hidden assumption, which is that the genome organization ought to be invariant cell to cell throughout the body, and so that you could allow mutation randomly, but the gene locations and linkages would stay the same. The basic structure of the genome should be the same in immunoglobulin secreting B cells as well as everything else. And so the question is this actually true? Well, at the beginning of this debate, there was no technology available to answer that question. And the best techniques at the time were really inconclusive. People used nucleic acid hybridization kinetics to try to count the genes. But the problem was that nucleic acid hybridization is affected by mismatch. And if you have imperfectly matched copies, because they're varied in sequence, the counting becomes very inaccurate. So when people developed these improved technologies that I was telling you about, restriction enzyme mapping and high-resolution size fractionation of DNA, it finally gave this field the tools that it needed to break this question open. And you could ask whether the sequences that were represented in the RNA that coded for the mature immunoglobulin were in contiguous units in the genome, and were they in the same place in all the cells of the body. And so this is actually a figure from the incredibly important paper by Hozumi and Tonegawa, which actually solved the problem in a very decisive way. And this is essentially a restriction mapping study to ask what sizes of genomic DNA actually include the sequences that are represented in the V region coding part of an immunoglobulin RNA and the constant region part of an immunoglobulin coding RNA. And when you cut up the genome with restriction enzymes, you get different sized pieces, and then the different parts of the molecule hybridize to different fragments. And this diagram shows what they found. That if you look at the size of the DNA from the genome of an embryo that hybridized with the V region part, it was a piece that was about 6,500 base pairs long. If you looked at the part of the genome from an embryo that hybridized with the C region part, it was about a 9 kilobase genome, and it wasn't the same fragment that included the V sequence. However, if you looked at DNA from the actual B cell that was producing this particular immunoglobulin, the two sequences were on the same sized restriction fragment. And it just happened that in this case, the restriction fragment was actually smaller than the V and the C together from the embryo. In other words, it wasn't just that someone took out a sequence that separated them. These had actually moved to be together. They were separated, and now they were together. And so this was dynamite. And that showed that for all of the different immunoglobulins, they also came from genes that were split in the embryo and together in the B cell. So essentially, something happened to physically change the organization of the genome in the B cell. Now, in this original paper, they actually laid out 4 different possible models for how this rearrangement could occur. We now know that it's a mixture of deletion and inversion in mice and humans, but that actually there are versions of this that are really like a copy choice mechanism that are used by rabbits and some other animals. So essentially, there's quite a few things going on. But the point was that you could have a special reaction, and this meant that first you no longer had this problem because you could now have multiple different V's, and as long as only one of them joined with the C, you basically had your diversity problem solved and your constancy problem solved. You had to postulate a new reaction. You could prove that if you could find the enzyme that mediated the reaction. And this was an explosion of work then. The Leader, Hood, and Tonegawa labs all got to work immediately to define what were the signal sequences that joined the pieces, that defined which pieces could be joined with each other. And these were all neck and neck over the next couple of years. Based on what they discovered, an assay was designed from these sequences that could then be used to score different types of cells to see which ones had an ability to carry out this recombination reaction. And it turned out that only immature immune cells had it. And this assay was used to clone genes for the ability to encode the recombinase activity. And this allowed rules to be discovered to explain then how immunoglobulin and T cell genes could be assembled. And so this was really a huge thing. I'm going to stop here because I was going to tell you a third story, but I've used up too much time. So I think that I'm going to skip then to the end and just say that this is about evolution. But I think that these discoveries changed our understanding in a very fundamental way. And I would say that they weren't Kuhnian because of the totally disparaging view that Kuhn had about normal science. that people are basically just hanging out, solving little trivial puzzles, which really don't advance the state of knowledge. This is these huge upheavals that break the normal science are not the only way that progress is made in science, and it is progress. Normal science in biology in the 1960s and 70s was in the process of developing rules, using them as working models to guide understanding and not fossilizing them into dogma, even though Crick liked to use this term. And the issues at the basis of future paradigm shifts had to be resolved by data, which could not even be collected yet at the beginning of this period, because new technologies had to be invented in order to observe them. Normal science at this time consisted of continuous innovation to create these technologies, specifically to enable new insights into these issues, not by accepting these things as being completely solved problems that simply had to have rules mechanically applied, but rather always an open frontier that needed constantly to be challenged with new approaches. And it was very important that people looked at different biological systems rather than a single unchanging universe in order to reveal information that would have been inaccessible if only a single system had been studied. I think that the impacts of these advances on the field were not incommensurable with previous paradigms, but instead they were straight out elaborations of the previous paradigms, which pointed out what was a generalization and what was just a common observation, which is a very big difference, in fact, in this situation. And the results transformed our understanding of the rules that govern biological systems. So I think that this is a case where the progress came from paradigm breaking that was within the scope of normal science and which was the mission of the normal science of the time. It was unbelievably exciting, it was extremely dynamic, it was enormously productive, and it gave us a radically transformed view of what the biological world is. And so I think I'll stop there. And if anyone wants me to go back and answer any questions about the part I didn't have a chance to show, I'm happy to do that. But otherwise, I'd be happy to take whatever questions there's time for.
So we have time for one quick question, and then we take a coffee break because we have additional 3 presentations. Yes, please. Ramon?
A great talk, Ellen. So can you mention one revolution in the immune field? In immunity, one revolution that happened that you can point that this is a revolution?
Well, I think that the understanding of how immunological diversity is generated was a bit of a revolution. I was going to talk about a different revolution, which was the revolution in understanding the evolution of immune receptors. But there wasn't time.
Thank you.
Okay, thank you, Ellen.
Unfortunately, this Zoom setup is not optimal for following the clock, and so, because I can't see presenter view from when I'm talking, so I apologize for poor time management.
It's okay. People can ask you questions via email or schedule a Zoom meeting with you. So thank you again. So we have a coffee break, and then we are back here 4:15.